Everybody that's logged in. I'm going to be moderating the session. So if you have any questions as Thomas is going through his presentation here, you can either type them in the chat or the Q&A section on the control panel there. If you want to actually join in the session and ask questions directly, I think there probably is still space for that. But if not, as I say, you can relay the questions through the chat function. And if you want to schedule a meeting based on what you've heard, then click on the schedule a meeting blue bar at the bottom of the table there, and that will let Thomas and Amy know that you're interested in a follow-up discussion, just a one-to-one discussion. So over to you, Thomas.
Right, thank you very much. So my name is Thomas Kendall. I'm an application specialist with Technobus crystallization systems. And today I'm going to be giving a presentation on co-crystals, of a perspective on the current practices, particularly around industry, and sort of how our products sort of integrate with the screening and crystallization development of co-crystals. So first, I'll just be touching on sort of an introduction into Technobus and the crystallization systems, before moving on to the more broad idea about the solid form landscape. Then touching on the salt co-crystal continuum, looking more at these sort of multi-component complex systems. So looking at the continuum, as well as sort of a definition of what is a salt, what is a co-crystal, and why that's important. Then moving on to sort of conducting a screen, so designing one initially, what to look for before moving on to sort of actually conducting and how you might do that. Then looking into scaling up of co-crystals, so how you can sort of control the co-crystallization process and get the most information and control out of it, and why that's important. I will touch briefly on formulation, so it's only really one slide, but it's something I don't see really gets talked about much with co-crystals, and it is an important aspect of trying to get co-crystals to market. And then finally talking about the benefits of co-crystals, which have been sort of widely discussed and sort of talked about, particularly say solubility and bioavailability, but I also wanted to touch on maybe a few others which haven't been as broadly covered. So what are Technobus crystallization systems? So what we are is a company that makes machines which allow platforms for accelerating crystallization research. So we've developed several unique and proprietary technologies which sort of aid in the adoption of research of crystallization and solid form. We have a lot of sort of instruments and made on agrochemical companies, and we have a big footprint in pharma as well, and fine chemicals. And our portfolio sort of contains 3 products which are based around different stages in the crystallization and solid form life cycle. So if you have a look at the drug discovery pipeline, and this may be just for drug discovery, but also for, say, agrochemicals, of where you have an early discovery of a molecule, you're starting out with, say, a couple hundred mgs of material. And as you go through the sort of process before reaching market, you get more material available, and you start to undertake more sort of screening. So you may look at salts, co-crystals, and polymorphs. Then moving on to crystallization, and then up to processing formulation optimization. And we have machines which sort of cater to those 3 areas. So you can use a crystal breeder for discovery when you have a small amount of material. The Crystal 16 when you're looking at, say, larger quantities and more looking at crystallization development, but as well looking at the solid form. And then optimization, so going even to higher volumes of material. So of the 3 products, we have the crystal breeder, which is 32 reactors, which is the sort of which is the largest you can go up to is 0.1 milliliter, so we're talking very small scale. The Crystal 16, which is 0.5 to 1 milliliter reactor, which is 16 reactors. And then the crystalline, which is 2.5 milliliters to 5 milliliters with 8 reactors. And all of these are multi-reactor systems with small footprints that are quite easy to use. All the reactors here are quite cheap and disposable. All our reactors have inline analytics, so there's no cleaning required after usage. and it's also small. So this is allowing you to get more information at the smaller scale. So when developing a drug product or any solid product, solid formulations are often selected due to the benefits offered to patients, such as ease of administration, lowest variability between dosages, overall stability of a compound, as well as sort of accurate dosing between differences between different tablets, as well as sort of convenience and ease of transport. But there's a whole range of solid forms that are available. So the question becomes, which solid form do you want to develop? And it sort of hinges on two different categories of factors. First, your biological factors. So how is it going to perform in the body through solubility, bioavailability, and dissolution rate? And then also economic factors. So how is it going to perform, say, in manufacturing? So its density, flowability, filterability, its drying properties. And you can find it can be sort of a compromise between the two, sort of finding one which is going to give you good biological results, so you can administer your drug to the patient, but also good economic factors, so it's actually going to be profitable in manufacture.
Thomas, can I ask a quick question about the regulatory aspect? So presumably there are a few co-crystals on the market now, but in terms of developing solid forms, is it as easy from a regulatory point of view to develop a co-crystal as it is other solid forms?
So I actually have a slide on that later on in the presentation. Short answer, it's the way the FDA classifies it as being similar to another polymorph. So it follows that regulatory, but I'll get into that in more detail later. So you have your molecule at the beginning, but obviously you can have different forms of this same molecule. You can introduce different, say, solvents, in which case you're getting solvates or hydrates being formed. You may look at different salts if you want to boost the aqueous solubility, as this is a typical way of doing it through salt formation. So you can have, say, chlorides or bromides, different salts being formed. And within that, you can have polymorphs of salts or even salt solvates. For co-crystals, we would then look at similar thing. You can use different co-formers to give you different co-crystals, and then polymorphs within those co-crystals. And then ultimately, you may look at, say, an amorphous solid dispersion where you're stabilizing the amorphous. And again, you can use different excipients to try and stabilize the amorphous. So just a visualization on what sort of these different forms sort of entail. You have your amorphous, which is characterized by its lack of order. And this is, in most cases, like a very unstable form and will ultimately crystallize to a polymorph unless it's stabilized. You can have different crystal packings of the same molecule, which give rise to polymorphs. So we have form 1 and form 2, just different packings of the same molecule. If we wanted to incorporate solvent or hydrates, we'd then be coming into solvates and hydrates, where you have this bridging almost effect, or the incorporation stabilizes the crystal structure. Before moving on to salts and co-crystals, where you introduce either a counterion or co-former to stabilize the crystal lattice. So what's the difference between salts and co-crystals? So because you can use the same molecule as counterion or co-former and get different results. So there's a reason you have malleate salts and maleic acid co-crystals. So to simplify it down, they exist on this sort of salt co-crystal continuum. And what defines whether you have a salt or co-crystal essentially comes down to whether you have proton transfer involved in the intermolecular bonding. So if you have a case where the hydrogen is not transferred, you start with this. You then have hydrogen bonding being the bonding motif, and that allows for the co-crystal to be formed. If you're then operating where the proton has been transferred, you're now beginning to have ionic bonding occurring, and then you start to form salts. And a good indicator from the outset to what might be being formed over a co-crystal or salt is the difference in the pKa's of the active and the co-former or counterion. So where we have a case of the delta pKa being less than 0, we have potential forming a co-crystal. Where we have it greater than 3, we're going to more likely form a salt. And it's in this region in between where you can get these sort of weird salt co-crystal hybrids, or even salt co-crystals where one end of a molecule is a salt and the other end is a co-crystal. It's not a sort of well-defined region in here. And this pKa rule is not, say, a hard and fast rule, but it's a good indicator of sort of what's going to potentially be formed. So to just sort of come back to the question, sort of the definition of what you have of your multi-component system of your form is important from a regulatory standpoint. As I said, co-crystals are, technically classified as different polymorphs, so they go down a polymorphic route for regulatory and testing. Where if you're going down a salt, it's a completely different route. So they are seen as completely different entities by, say, the FDA, which is the major sort of drug definition group in America. And they define it as a crystalline material comprised of two or more different molecules, one which is the API in a defined stoichiometric ratio with the same crystal lattice that are associated by non-ionic and non-covalent bonds. So again, going to the idea of, they are, say, hydrogen bonding, of a neutral species, which isn't a solvent. And it's classified by this as being a drug product intermediate. So once the co-crystal enters the body, it's believed that it dissociates into co-crystal and co-former, into a active and co-former, and then this active is then treated as being the primary molecule, as if it was just a regular polymorph. So identifying what you have, so new crystalline entities can be identified by powder X-ray diffraction, but to sort of, and DSC, and that will give you an idea of, say, if there's a new unique melt, that can be a good indicator of a new co-crystal. And solid state NMR can also be used to sort of determine if you have a salt or a co-crystal. However, single crystal X-ray diffraction is really the only definitive proof of whether a co-crystal or a salt has been formed. And if you're doing, say, filings for a salt or co-crystal, this would be highly recommended for information to include with any patent applications. So how might you go about sort of designing and conducting a co-crystal screen? So predicting what bonding interactions currently is very challenging. There's no sort of software that will allow you to do this. But using crystal engineering and sort of a database of crystal structures, something like the CCDC, it's able to look at what bonding structures what bonding patterns and motifs exist within that molecule. And that could be a strong indicator as to what bonding patterns could be used, and then co-formers could be used to sort of recreate those bonding patterns. And something using sort of called supramolecular symphons. So symphons diamonds where you've got, say, the same molecule bonding within the crystal. So in this case, we have carboxylic acids, or you might look at ones where you have, say, different functional groups, carboxylic acid and pyridines and amide diamonds, or you sort of have this carboxylic acid bonding to the different functional group. And again, this isn't a hard and fast rule. It's not going to give you exactly knowledge of where the bonding is going to be occurring. There's been several cases of where I've designed a co-crystal screen, looked at it and thought that's where the bonding site is, only for the single crystal to come back and be completely not what I expected. It's not even involved in the bonding structure at all.
Thomas, can I ask a quick question about the structure of the co-former? So is there an accepted kind of database of molecules that you can use as co-crystals? Because I guess the toxicity of the co-former is potentially an issue as well.
Absolutely. So there is a list of FDA-approved co-formers. I believe it's on their website, and there's, like, I think it goes up to like 100. There's quite a lot in there, and they will actually give you the amount is that a person can consume within, say, a 1-day period. So you can sort of have an idea of how much your dosage will be based on that. So using the crystal structure database, it's possible to see favored by the molecule or by structurally similar molecules. So once you've sort of co-formers and you've sort of decided you want to start doing a co-crystal screen, use a sort of range of techniques. And similar to polymorphism, a lot of the techniques can be similar to what you use in a polymorphism screen. So you'd look at liquid-assisted grinding. So by creating interactions between two steel balls with solvent in there, you can help sort of ripen in between those high-impact interactions. Evaporation, by driving off the solvent, you sort of generate your supersaturation, take out your co-crystal. Cooling, so again, decreasing the temperature and then driving supersaturation. Anti-solvent, by changing the solubility of the solvent system completely, or through slurring by ripening the solids in suspension. And these techniques can be used in a combination, say varying stoichiometries of active ingredient co-former to be able to produce a phase pure co-crystal. You want to try and hit as many different crystallization routes and solvent functionalities as possible. There's only certain co-crystals will only be formed using certain techniques. So this is an example where solvent drop grinding is giving you loads of hits, where evaporation is only giving you a limited number. And similar thing with anti-solvent, you're getting even less from that. So having a wide range and using as many different techniques is recommended. However, it's important to note that when you're coming to scale up of a co-crystal, the only real ones that can be scaled up to large scale is through solution-based. There are significant advancements in solid state co-crystallization at scale and continuous through hot melt extrusion or through continuous milling, but they're not nearly at the stage of where you could take it to, say, an industrial scale. So you would need to look at solution-based routes during any co-crystal screening to identify a way of doing that. So what you can do is employ a more systematic approach to co-crystal screening, utilizing the solubility measurements that you can make to miss co-crystals, and then take that information and carry it forward to the scale-up phase. And why might you miss a co-crystal potentially being formed? Where, say, if you've got a one-to-one stoichiometry and this is a nice behaving co-crystal system, you're happily going to sit inside this phase diagram region here, AB, which is the co-crystal of component B and A. So it's quite nice and happy sitting in there, and you'll be able to find the co-crystal easily. Alternatively, if the solubilities of the two components are wildly different, you might actually be by going for a one-to-one stoichiometry, but be missing entirely and only be getting your component B, or having your co-crystal and extra of component B being formed. So you can potentially miss that out, but just by going blind in with a one-to-one stoichiometric ratio. So how do you collect solubility? So there's three main ways of doing this. So first would be equilibrium concentration, which is the most accurate way of doing it. However, this is very time consuming. So slurring material, extracting the mother liquors, and then analyzing the mother liquor through, say, HPLC. But this requires a calibration. And as I said, it's very time consuming and only really gives you one temperature point, sorry. An alternative would be to do solvent addition. So this is taking a known amount of material and adding solvent in aliquots until you get to solution. Again, this is very time consuming and can only give you really one temperature point. The alternative is using a full solubility temperature variation model. And this is where you make several concentrations and measure when the solution is obtained. So when the material goes into solution at various temperatures, and that can help you generate a solubility curve. So this is done using transmissivity. So all our machines have this technology of where we have a laser light going through your sample. And when there's material on there, the laser light is obscured. So then we have a low transmissivity. And when it goes clear, we then reach 100%, which is clear transmissivity. And this is an example of sort of the data you generate. Of the red is the temperature profile. And in blue, we have the transmissivity. So where we suddenly go from 0 to 100, we've now gone clear, and this is our solubility point. So by collecting across multiple different concentrations, we're able to generate a solubility curve, which you can then extrapolate data from. So the way you would conduct these sort of systematic approaches to co-crystallization is by collecting solubility of the pure components, and then creating a solid mixture of those two components and finding the solubility temperature again. looking at what you formed, so XRPD, to confirm the formation of a new phase, and then you can begin sort of construction of a phase diagram before moving on to single crystal preparation or scale up. So you would collect the solubility curve of the individual components here, and as you can see, it's quite a broad distance between the two. You would then identify a temperature where you would want to measure, say, the solubility of the components together. So you would take something like, in this case, 32 mig per mole, millimole per mole, sorry, and here about 7.5 or 5. You would then create a mixture of these two components together, slurry them at 25 degrees C and start heating. And if the solubility point is greater than 25, the solubility then is no longer dictated by these two components, and it's more likely to be a less soluble co-crystal phase. So one where if you've got a new co-crystal being formed by having a higher solubility, you would then collect a solubility point across a range of variable sort of coformer against active, so A over XA XB. And then where you find the region that the solubility is dictated by the co-crystal, this would be a not safe operation region to sort of develop a co-crystal, or at least seed during scale up. And if you've got a one-to-one, this can be quite a nice region. But if you've got something that's maybe a bit more skewed, this can sort of give you a quite difficult region to operate in. But this sort of information is very important when it comes to scaling up or developing single crystal experiments. So moving on to crystallization. So the benefits of crystallization: it allows you to control your solid form through seeding protocols by introducing the known solid forms you want to introduce. Allows you to influence particle size and shape by changing the supersaturation generation through cooling and anti-solvent profiles, or even by adding additives. You're able to sort of encourage the growth on certain crystal faces, influencing morphology. It also allows you to increase your purity by purging impurities that might not be able to be purged through the chemistry. And a controlled crystallization is necessary in any manufacturing and legal filing of active compounds. So having a good controlled one, and I think sort of discussed earlier today in some of the presentations, you have to go through that GMP manufacturing, which requires a whole load of legal filings with it. So having controlled crystallization is a necessity. And the crystallization itself is a complex process consisting of multi-component competing factors and processes. So your compound, the solvent system, the thermodynamics and kinetics of the system, so your temperature, if there's different forms or phases present, as well as the process, so the scale of experiment, order of operations. Do you want to cool first and then add anti-solvent, or add anti-solvent and then cool? And these can give wildly different results. Stirring and the type of impeller, and I think shown today earlier in some of the presentations, even the type of impeller has a big impact on, say, the results of the crystallization by effect of, say, agglomeration and breakage. So crystallization itself is a complex process by itself. But then when you're introducing a co-crystallization, it sort of increases the level of complexity, because now you have two individual components and nucleate spontaneously by themselves, giving you phase and pure material. Therefore, it's important to employ several techniques to control the crystallization process. So this can be done using process analytical technology, so PAT, such as UV or IR, sort of tracking the supersaturation and concentration in solution, using optical microscopy, such as PVM, sort of being able to see what's happening inside your sample, using chord length measurements to sort of get an idea about morphology and size, as well as Raman to maybe identify form purity. Designing a seeding strategy to ensure you're crystallizing the desired form rather than allowing it to be spontaneously crystallizing, which might give you unwanted forms or polymorphs. Phase diagram to understand the safe region to operate. So are you going to be sort of treading in a dangerous region where you might start to get spontaneously nucleating one of your other components? And using modeling software to get the most information of the data collected. And there have been several case studies of using this, using a combination of these techniques to produce robust, scalable co-crystallization in both batch and continuous. So this is an example of sort of what you can collect using the PAT. So this is collected on our crystalline instrument. So this is at 2.5 milliliters. We're able to see real-time phase transitions. So we can have sort of the onset of crystallization occurring as they start to agglomerate, whether you're forming a foam, so this is tiny bubbles being formed, or even oiling out. And all of these sort of have major implications on the crystallization development. Another one, so again, using our crystalline, we're able to integrate Raman. And this is in being able to track the formation of different forms. So in this case, they have form A and form E, and they're being able to perform form B. And they wanted to understand why this was being formed, or what controls for this hydrated form B were sort of, when did this start to appear? And the principle is the same for co-crystals. If a co-crystal is a unique Raman spectrum or peak, you're able to track that in real time. So they've did a series of experiments to understand where you start to get the formation of B by varying the water activity in percentage. And they found that sort of around 3 to 4%, you start to get B creeping in. And then by 6%, you have a full conversion to the hydrate. So this sort of gave an idea about the storage of their material, as well as designing a crystallization to avoid the formation of B. As I said, you can track this in real time. So you have form A being formed, and eventually over time this converts a decrease in a specific peak, and then an increase in your form B peak. And you're able to track then the formation of your co-crystal, in this case form B, in real time. So just an example of being able to pull these understandings of the solid form landscape and pulling PAT controls. Is a case study of a caffeine glutamic acid co-crystal, which was scaled up to 1 liter. So there's 2 known polymorphs of this co-crystal, form 1, which is the kinetic metastable form, and form 2, which is the stable thermodynamic form. And through studying the cooling rate and seed loadings, they were able to find that a high level of supersaturation led to secondary spontaneous nucleation of the undesired kinetic form 1. So by implementing the understanding the solubility curve, by implementing the understanding the concentration in solution using an ATR FTIR probe and PVM, they are able to design a co-crystallization process with high C loading and a slow cooling rate to ensure that they're not generating too much supersaturation, and they're focusing on growing crystals rather than spontaneously nucleating the undesired polymorph. So formulation of co-crystals. So during drug development, formulation consists of multiple processes, including milling and utilizes several excipients to aid in the acceptability and solubility, and ensuring that the drug can sort of be delivered to the patient at point of use. However, in a lot of these cases, the excipients can result in dissociation of the co-crystal. So a study on the reported stable caffeine oxalic acid co-crystal found that dissociation would occur during the formulation process with commonly FDA-approved excipients. And they found that it related to the acid-base nature of the excipient. So you have the excipient, which has its hydrogen bonding characteristics, and these would provide alternatives to the co-crystal. So you would have dissociation by providing alternative bonding sites. So it's important here to say, consider what excipient you're using. Additionally, they found that in a different study of a theophylline glutaric acid co-crystal, that a high hygroscopicity and high ionizability resulted in significant dissociation of the co-crystal, where a low hygroscopicity excipient showed no dissociation after being stored at elevated conditions. So it sort of emphasizes the importance of looking at what excipients are using and what effect that can then have on the co-crystal being stability. And finally, I wanted to touch on the benefits of co-crystals. So this has been sort of widely talked about for a while, and as the publication on year increases, you start to see these sort of same trends of an improvement in solubility and bioavailability, But what I wanted to touch on maybe a few different ones, so taste masking or chiral resolution. So I'll get the solubility ones out the way sort of early, and that's sort of the way you would maybe improve the solubility is by, say, salt formation. But if you don't have an ionizable center, you may want to look at co-crystals. So this is aripiprazole. It's a atypical antipsychotic used in the treatment of schizophrenia. And a co-crystal with fumaric acid was discovered, which improved the thermal stability and solubility with a rapid onset improvement in the bioavailability. Solubility, so this can be also the opposite can be true. So if you have too high a solubility, it doesn't provide the required therapeutic profile. So it goes into the body too quickly and is processed too quickly. Co-crystals can be used to modulate that by giving a sustained, slower release. In this case, we have zionzamide, which is an anticonvulsant, which has a high solubility and dissolution profile. So several co-crystals were made using various different co-formers and was tested with IDR, which is the intrinsic dissolution rate. And caffeine was found to have the biggest improvement in dissolution profile, opening up the potential for a sustained release of this drug. A sort of benefit of co-crystals, you're able to sort of combine two actives together. So this is a dual action. What we have is saloxalib, which is a COX-2 inhibitor, a non-steroidal anti-inflammatory, and tramadol, which is an opioid pain medication. A one-to-one co-crystal of this was made, and a dissolution rate showed that the saloxalib was found to increase, while the tramadol was found to decrease, which would indicate a reduced near side effects of the tramadol. So this showed a benefit over the monotherapies of both. So a dual action formulation is open to this. Permeability. So dermal administration is an effective delivery method for some treatments, and it can improve the patient's compliance and with less frequently dosage. However, molecule has to have good skin permeability, which isn't always the case. Here we have a case of 5-fluorosil, which is used in the treatment of skin cancer by topical application. However, it's limited due to its poor permeability and skin penetration. They did a series of studies on co-formers of benzoic acid-derived co-formers, followed by single crystal analysis to understand what bonding motifs were involved in the formation of the co-crystal, as well as permeability, and they found improved permeability with more lipophilic co-formers. They then selected a highly lipophilic co-former, 6-hydroxy-2-naphthenic acid, which was then tested and showed a significant improvement in permeability compared to the API by itself using a Fran cell. Stability. So co-crystals can offer a unique way of improving the stability of molecule by protecting sites which may be susceptible to oxidation or hydrolysis. So if you're able to bond a co-former directly or indirectly, blocking off that molecule, that part of the molecule which is, say, susceptible, you're able to protect it. And there's a case where brexpiprazole is used in the treatment of antipsychotic disorders. It was found to undergo photooxidation of the nitrogen in the pyrazine ring during granulation with PVP. It was found that during the PPV manufacturing, there was residual peroxides left, and this was resulting in, these were attacking the molecule. So they were able to form a co-crystal of ketracol, which is shown here, bonding directly with the molecule nitrogen involved in the photooxidation, preventing it from being broken. Taste masking. So taste has been shown to be an important factor in the acceptability and willingness of a patient to take the drugs, especially in children, adolescents, and the elderly. Co-crystals can be formed with sweeteners and taste maskers as co-formers, which have been shown to improve the taste of the drug product. As well as an added benefit is a lot of these sweeteners and taste maskers are already FDA approved for application in food. So theophylline is a drug used in the treatment of therapy of respiratory disease. And they formed a co-crystal with saccharin, so the artificial sweetener. And it was found to improve the drug release profile as a suspension. as well as improving the overall taste and masking the bitterness on taste sensor measurements. And then finally, chiral resolution. So chiral purity is an important factor in the effectiveness of a drug, where one enantiomer can be medicine for a disease and another one can be, in fact, toxic to a patient. So chiral resolution is typically done either through the chemistry or through recrystallization using resolving reagents, and these are typically salts. However, if you don't have a ionizable center, you can't really go through this route. So an alternative, which is being investigated more increasingly, is the use of coke formers as resolving agents. So you can do these rapid screening tests using sort of the processes outlined previously, and then followed by chiral resolution to see if you've been able to refine your chiral resolution. And an example here on large scale is ilamivudine, an antiretroviral, which was separated from its enantiomer using S-binol at 30 kilograms, and then was allowed purified to give a 99% enantiomeric excess. So being able to use an alternative to salts for chiral resolution. So thank you for listening to my presentation. Just as quick on Technobus, we have webinars every month on crystallization and solid form topics. We have application notes on a range of topics such as solubility, solid state, process development, formulations. And if you have any questions sort of related to applications or about our instruments, please please email at info@crystallizationsystems.com. And we're also celebrating 16 years of Crystal 16, which will be in September, where we'll be holding a series of workshops around crystallization and solid form development. Thank you.
Well, thank you, Thomas. That was really interesting, actually. I have a couple of questions about. So, is the definition of a co-crystal? When does a co-crystallizer become an impurity? Does it have to be within a defined kind of stoichiometry to be regarded as a co-crystallizer?
I think, yeah, it has to be sort of a reproducible stoichiometry. I don't think there's really a case of where it would be classified as an impurity. Maybe if there's, it varies, or if it's found to be. No, I can't think of where it would be classified as an impurity, really.
Okay. It's something to do with has to be in the unit cell of the crystal, is that right? And,
yes, so it has to be involved sort of directly bonding non-covalently or ionically with the parent molecule.
Okay. And do you do, Technobus can do screening for co-crystals, or do you just sell the equipment to do it, or can you actually do it yourselves for other people?
No, so we sell the sort of the instrument which will allow you to do that. So you can do sort of within the Crystal 16 or Crystal Breeder, various temperature profiles, slurrying, cooling, sort of evaporation or anti-solvent addition.
Okay. And then finally, the kind of scale-up aspect. I know you kind of mentioned that, but what are some of the challenges going from once you've identified a co-crystal, actually going into kind of making that on large scale or even manufacturing? Is there a level of complexity above and beyond kind of normal crystallization? And how do you actually control when you do the crystallization, the actual stoichiometry?
That's a very good question, and you sort of need to get as much information at the low level as you can.
Okay.
Sort of build up as much information as you can at, say, the lower levels, and as you go up gradually. You can't just say jump straight from 1 milliliter to 100 milliliters and expect it to go completely fine. Build up the understanding as you go, because as you go up and up, the level of control is less. So whilst you have high level of control, start understanding your design parameters, start testing the limits, do a DOE to understand, if I push this profile a bit too far, what happens? If I go below this level, what happens? And sort of get all that information at the lower scale before going up.
Okay. And do they have separate identities in terms of kind of cast numbers and things? So a co-crystal of an API, for instance, is it regarded as a different chemical entity?
That's a good question. I've never actually thought about that, and I haven't looked. As far to my knowledge, it doesn't. It's just listed as a co-crystal of that cast number.
Yeah, it's just like a salt, isn't it? I guess it's just a different form of the API.
Yeah, it's sort of that definition by the FDA is kind of weird because it opens up, I feel, quite a lot of, you know, worms of. So I have if I'm an innovator and I don't do a co-crystal screen, am I now liable to being undermined by generics who find a co-crystal? Yeah. Which is what the FDA has essentially said in their statements.
Okay. We have a question that's just come into the chat. So, how do you avoid evaporation in crystal 16 vials? So, this person's found that it can be a challenge for low boiling point solvents, especially with overhead stirring. Any comments on that?
So with the overhead stirring, there should be sort of O-rings that allow you to sort of seal it up tighter than sort of just regular screw cap. I would recommend going, say, 5 degrees below the boiling point of your solvent, just to avoid, you know, potential evaporation occurring, as well as maybe operating at the 1 milliliter volume. I don't know if, Amy, you have any more comments?
I do. I also have a little bit of advice. What we're seeing, if you're buying vials from a commercial vendor, such as Fisher or VWR, sometimes the glassware tolerance is an issue. So that may be one solution. I'll touch base with you as well. But what we see is sometimes the threads and the tops, they're not quite in spec as they should be when you're going, again, through the vendor Fisher VWR on the throwaway disposable vials.
Okay. Great. Is anyone else that's in the chat want to ask any questions, or do they use the Crystal 16 and want to ask any questions? Because we've got the world experts in the room here, so it's a good chance to ask any questions that you might have.
And certainly also, please feel free to contact us. We can work with you, especially on some of these applications. We have so much material out there that can really provide guidance.
So what's the next innovation in Crystal 16 design? Have you got any new prototypes in your thing? Or
we do have product development going on. We have some really great things coming out. I can't make any announcements, but stay tuned would be the message I have.
Great. Okay, and then you have the Crystal 16. You have another system, don't you, that's slightly larger, is that right?
Yes, so we actually have 3. We start with our crystal breeder, which is really polymorph screening, just single crystal growth. The Crystal 16, which I think almost everybody on here probably has one, and that's really day in, day out, solubility curves, metastable zone width. And then we go to our crystalline, where we can actually see at a slightly larger volume what's happening. We add cameras and we add Raman as well.
Okay.
And again, I look forward to when we can do this again in person and really have these on the bench again. I know, John, we've been at so many of these conferences over the years that, yeah,
it will be good. So do you get good translation from the Crystal 16 to the crystalline? Do you get very effective scale up from the smaller to the larger unit generally?
We do. Thomas can probably talk more about it, but I do know we also have publications and white papers on this that are on our website as well.
Yeah, absolutely. We do get great sort of translation. Obviously, the mixing dynamics change as you go up in scale. Yeah. But for, say, solubility points, you're pretty spot on, even up to 1 liter scale. That's sort of, the benefit of having solubility, which is a thermodynamic property. It's translatable really well throughout the system.
Okay. We have another question about, can you add PAT probes to Crystal 16? I guess there's not a lot of space in there for a probe, but
yeah, the Crystal 16 typically not. I'm not seeing it, but that's where the crystalline comes from. It's really the next step. So you create your roadmap with the Crystal 16, and as we move into the crystalline, then we do have the added analytical tools. So we have the cameras that are giving particle size measurement, particle size distribution, or we're able to use it with Raman. I've had people with the crystalline also stick IR probes into the vials and other types of, basically inline technology.
Yeah, and if you think about what the first scale where the commercially available PAT tools are acceptable, say 100 milliliters, being able to do this at 5 milliliters adds a lot of information at lower scale. Also, just maybe to touch on additional things people have used, we know people have applied pH probes as well. So if you're say, looking at protein crystallization, which can be quite pH driven, that's something as well we've seen people use in applications.
okay. And the crystalline, I guess it has overhead agitators, which is good.
Yeah, both the crystal, actually all three of the products, we do have the option of overhead stirring.
Okay. And in terms of the stirrer configuration, can you get different stirrer types? I guess
you can. We have, and I always make jokes about my chemists enjoy and use more our dough hook design. Whereas my chemical engineers, we have 3-blade traditional impellers that they tend to stick more with that type of a stirrer design.
Okay. I guess everybody has their favorites, don't they?
They do.
Okay, we have just a comment that someone's using Teflon tape and parafilm to seal up their vials. So maybe that, I'm not sure how reliable that would be, but obviously works for them. So very good. Okay, I think co-crystal area is quite an interesting area. Actually, it's I've worked on one co-crystal in my past, and we did have some problems trying to scale it up. That was really the origin of my question, is just controlling the stoichiometry. We had to use some kind of melt extruder or something, I think, in the end to try and figure it out. But yeah, this looks like a nice platform for at least screening for co-formers.
Yeah, if I could do maybe a bit of self-promotion, I do have a book chapter which I wrote with Johnson Maffey, who are giving the talk opposite us right now, when I previously worked there, and it's on an industrial perspective of co-crystals, so it has more detail about scale-up formulation, screening, and sort of regulatory, especially legal aspects we cover quite broadly as well.
Okay. So how do the kind of patent authorities view co-crystals? So if a generic company, because I know the whole kind of polymorph thing has been it's a little bit more controversial now than it used to be. I think, generic companies could patent a different polymorph, but I'm not sure it's that clear cut now, certainly in certain, jurisdictions and certain patent groups.
Absolutely, and you also have to patent how you make it as well.
Right.
I think it's the way they've classified it as being a separate crystal entity. It does open up an entire strategy of being able, of a generic, to just come up with a co-crystal, and that's the new one. It's a completely different protected entity.
Okay.
As long as you're within, say, the freedom to operate.
Yeah.
I think there's several cases, even in agro, you form a co-crystal of it. Even in CBD, there's co-crystals of CBD, which is just undermining, the solid form of certain cannabinoids.
Yeah. And do you find that people are screening for co-crystals now as part of the typical kind of solid form screening that you do at an early stage? So you might do like a polymorph screen on a new molecule, for instance, and you just run a co-crystal screen at the same time, or?
So I can only speak from my previous experience and my previous employment at a CRO. but we did see more co-crystal screens sort of coming in as time went on.
Yeah.
And I think, you know, pharma is very slow to adopt new things, so it still is kind of set in its old ways. But eventually, co-crystals will be accepted as widely as salts. So it's something I think a lot of companies will just have to do as part of their normal polymorphic work.
I guess the difference is people can kind of rationally come up potentially with a range of salts to screen, but with co-crystals, it's very much purely a kind of empirical screening exercise, isn't it? I guess maybe the in silico kind of modeling side of things will, in the future, make it easier to kind of select some of these things.
Absolutely. As you look at databases of what co-crystals exist, you start to get an idea of what bond, as I showed with the synthons, what bonding motifs are consistent, what keeps reappearing.
Yeah.
If you see that in your molecule, you go, okay, that could potentially be somewhere someone could come in. But the modeling isn't at the stage where it can do that yet.
Yeah.
In the same way you can't predict the number of polymorphs or solubility. But it's getting there.
And do you have any feel about the level of polymorphism in co-crystals? Is it the same kind of what you'd expect in normal kind of APIs, or?
I think so. It's because the number of polymorphs can be like, you know, it ranges quite broadly depending on, say, how many bonding sites there are, how large the molecule is, how flexible it is. Number of polymorphs have a high number of co-crystals, and if it has that same bonding site, but you can just tweak the molecule into a different configuration and maintain a crystal structure,
yeah,
you're still going to get multiple polymorphs.
Yeah.
And then you add like the level of complexity of, if you have a coformer, which is as well very flexible, you have polymorphs of the which are potentially from the coformer being different configuration.
Yeah. Okay. Very good. So if anybody has any more questions, I think we're probably going to be rounding up the session. Are you guys going to stay in the expo for a little bit, or what are your plans now?
Yep, absolutely. We will have in our basically expo booth virtually. Yeah. So if anybody has questions or need anything, let us know. Also, if you just want to comment, if you would like a recording of this, I know Claire said she will be providing a recording, so I would be very happy to make sure you receive it.
And then if on the basis of what you've heard, you want to have a kind of one-to-one discussion with Technobus, you can just go into the expo and just request, just click on that request further information, and Amy and her team will contact you, and you can talk a little bit more about your specific requirements. If it's not something you want to do publicly, you can do it in a more controlled environment, should I say.
Absolutely.
Brilliant. Okay, well, that's fascinating. I've learned quite a bit this afternoon, so thank you for that. And well, if people want to move over to the expo, they can have more of a chat with you, more informal, I guess. And I hope you enjoy the rest of the conference tomorrow, and PAT if you're joining us for PAT on Wednesday. So thank you.
Thank you, John.
Thank you,
All right. Take care.
Bye-bye.
Bye.
Bye.